Hi guys, my name is Anna, and in this video we're going to discuss the process of the cell cycle followed by mitosis. Please grab a piece of paper so you can take the notes alongside me, and by the end of this video you're going to be experts and you will know everything you need to know for A-Level Biology. The only thing I ask in return is please press the subscribe button and like this video, and do drop me a comment below on what other topics you'd like to see. And without further ado, let's get into this topic. Before we can go through mitosis, it's important to learn about the process of the cell cycle. So let's draw a beautiful big circle and name the stages. So the first one is going to be the G1 phase, standing for the growth. Then it's followed by the S phase, which stands for the synthesis phase, and then it's followed by G2 phase and mitosis. Quite often, it's forgotten that mitosis is straight away followed by the process of cytokinesis, which we're going to discuss in a moment, but it's important to understand that it's actually separate to mitosis itself. Now let's go through each one of these stages and understand what goes on in each one of those. So let's start with the G1 phase. As I already mentioned to you before, we refer to it as a growth or gap phase. And in this stage, basically cells grow and there are going to be new organelles that are going to be made, which will then end up in future daughter cells. The next stage is S phase or synthesis phase. And during this stage, the DNA is synthesized, and it's basically the same process that can be referred as DNA replication. Then in the G2 phase, which is basically the second growth phase or GAP2 phase. And at this point, there is a lot of protein synthesis happening and the production of different micromolecules. The G2 phase is then followed by mitosis, which is the actual process of cell division. And then cytokinesis is the process that happens after mitosis, and it's the physical separation of divided cells. The cell cycle is a highly regulated process, and therefore the cells have evolved to have checkpoints across the cell cycle. So there is a first checkpoint referred to as G1 checkpoint before the S phase, and as you can guess, it's going to be there to check that the cell is first of all ready for DNA replication and that there are no mistakes or damage within the DNA. So then the cell is going to be ready for the S phase. The second checkpoint is called G2 checkpoint, and it's between the G2 phase and mitosis. And it's basically there to check that all of the DNA has been replicated correctly without damage at the S phase. So it's basically preparing it for mitosis and saying, okay, well, it's a green light to go ahead and for the cell to actually divide and to produce daughter cells. So as you can appreciate, these checkpoints are quite important. There is going to be a third checkpoint during mitosis, but we'll get into it in a moment. The last thing you need to know about the cell cycle is that collectively G1 phase, S phase, together with the G2 phase are called the interphase. So as you can see, I've now labeled this with orange, leaving the mitosis and cytokinesis out and labeling this as an interphase. So it's just the state the cell is in when it's not undergoing mitosis. So let's quickly discuss how the cell might look in the interphase. So if you guys draw a round cell here, and we're going to basically add some features to it, which you can then describe. So first of all, the cell in the interphase, as a reminder, is going to be at the stage of G1, S phase, and G2 collectively. The interphase cell will have a nuclear envelope, which I'm drawing here, with nuclear pores present as always. So those are the gaps representing that feature. And then there will be DNA, obviously, inside of the nuclear envelope. And the way the DNA looks, it's basically not going to be condensed. Remember, guys, the DNA is only visible during mitosis when the DNA actually does become condensed, but more about this later. Right, let's also label the presence of the nuclear envelope. And then the third feature that represents interphase, or the cell in the interphase, is the presence of the organelle called centriole. And centrioles, if we draw them like little L shapes and stars, they're going to be present in pairs. The function of centrioles is that they act as centers from where the microtubules grow. So microtubules will be forming the spindle fibers during mitosis, so you can see how the centrioles will be very important, which we'll discuss further in detail, and their role for mitosis. So this is how the interphase cell looks here. Now let's jump onto the process of mitosis and discuss what happens at each stage. So the way we're going to separate it is we're obviously going to divide it into 4 stages, and the 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. And we can now go through each of these stages, and it's very important to be able to draw them out. So let's start with the first stage of mitosis called prophase. Let's first start with drawing centrioles, again as little L shapes and stars around. And as you can see now, the 2 centrioles that used to be together during the interphase stage, they're now separated and they have migrated to the different poles of the cell. And this is where the spindle fibers will grow and will be formed. So let's now add some spindle fibers growing from centrioles. Also add some chromosomes to the diagram. They will be represented as crosses as they are after the DNA replication. Please watch my meiosis video to have that explained if you're not quite sure. So let's add to our note. So spindle fibers are formed, and then the chromosomes are condensed at this stage. So what it means is that they can be observed through the mitosis. They kind of shorten and become slightly wider. And also, guys, note there is basically no nuclear envelope present at this stage. So what I also want to kind of change here as well, to make it slightly different from the next stage, I guys want you just to kind of randomize the chromosomes and their location quite a bit, not to confuse it with metaphase. This brings us to the second stage of mitosis called metaphase. So we're going to draw a cell, we're going to draw our centrioles at the opposite poles of that cell, and we're going to add spindle fibers growing from those 2 centrioles. And the presence of chromosomes right in the middle is very important and actually is indicative for the stage of the metaphase. If you're struggling to remember all these names, you could remember middle M for metaphase. So here, the chromosomes are lined up right in the middle of the cell, which in the cell cycle terminology is called an equator. Also, during metaphase, the spindle fibers actually attach to the parts of the chromosomes called centromeres. And just to clarify, so the centromeres will be present in the middle of that cross. Metaphase is also an important stage of mitosis because it provides an extra checkpoint called metaphase checkpoint. And it basically ensures that these spindle fibers have properly attached to the centromeres. Because you can imagine that if the spindle fiber hasn't attached properly to a chromosome, when the cell pulls them apart in a moment to separate poles, it actually may result in the cell having the wrong number of chromosomes then eventually present in the daughter cell. This brings us to the third stage of mitosis called anaphase. So if we draw a cell again with 2 centrioles present at the opposite poles as in the previous cells, but the difference here is going to be is that the anaphase is the stage where the chromosomes, or otherwise we now need to start referring to them as sister chromatids, which are being separated. And this is the key point, guys, that in mitosis, in exam, you have to say that sister chromatids are being pulled towards different poles. And you can see that in this case, the spindle fibers, they actually contract and they are undergoing the shortening. And then it's the chromatids that are being pulled apart to the separate poles. And as I even underlined it here just to kind of highlight the importance of the phrasing for exam. And then this brings us to the final stage of mitosis called telophase. And here it's important to almost draw 2 cells attached together, but it's important to understand that actually those cells haven't physically undergone separation yet. So during telophase, the chromosomes uncondense, so they become thinner and longer as in interphase. And also what you can see, guys, what I've drawn here is that the nuclear envelope begins to reform. So the cell kind of starts looking a little bit more as it used to during the interphase stages, although we now have these two cells that are still attached to each other. So you need to remember that telophase is the last stage of mitosis, and cytokinesis is actually not part of mitosis, but it's an essential stage that actually results in a physical separation of cells. So now, eventually, we have the product of mitosis, which is two daughter cells. The last thing I want to mention that can come up in certain exams is the idea of mitotic index. So some specifications require you to learn this equation, but sometimes it might be given to you in exam. So please do check what your exam board requires. But basically the idea is, it's quite a simple forward equation, which says that mitotic index equals to cells in mitosis divided by the total number of cells. So the skill here is, and it usually can come up in assessed practical, is that you need to be able to identify cell in mitosis. And how do we do this? It's by seeing those chromosomes that are condensed, because you're not going to see chromosomes with the light microscope if the cell is not in mitosis. So for example, let's say a question asks, okay, so you identify 10 cells out of 100 that are in mitosis. Therefore, the mitotic index is going to be 0.1. This brings us to the end of this video. I hope you enjoyed it and learned loads of useful things. Please press the subscribe and like this video, and do let me know what is the next topic you'd like to see in my videos. And thank you for watching. See you in the next video. Goodbye.